want to welcome everyone, Duke alumni and friends, to this session of the Duke Alumni Forever Learning Institute. It's an interdisciplinary educational program organized in a set of thematic courses. And my name is Matt Hunter. I'm Assistant Vice President of Duke Science and Technology. And I want to thank you all for joining our program, Nurturing Nature, Improving Health with Biomaterials. This program is part of our theme, Material Sciences Designing a Better World, and these courses will run throughout the month of May. Moderating our discussion tonight is Professor Stefan Zaucher. He's a professor of mechanical engineering and material science and director of the Duke Materials Initiative. Welcome to the Forever Learning Institute. And Stefan, I will turn it over to you.
So I'm excited to welcome you to today's session, Nurturing Nature, Improving Health with Biomaterials, in FLI's Spring Lecture Series of Material Science. As Matt pointed out, I'm Stefan Zaucher, Professor of Mechanical Engineering and Materials Science, and also Director of the Duke Materials Initiative. And DMI is co-sponsoring these lectures, and I would like to give you a brief glimpse into DMI's exciting activities. First and foremost, DMI is the home of a growing collaboration of faculty and researchers in the broad field of material science and engineering across Duke University. We established the Duke Materials Initiative in 2019 to create materials through bold and also transformative advances that are really only possible when we cross and extend beyond the boundaries of traditional departmental disciplines. And I would add also our campus gates. And if you look at this graphic, we established the Duke Materials Initiative in 2019 to create materials through bold and also transformative advances that are really only possible when we cross and extend beyond the boundaries of traditional departmental disciplines. And also, I would add our campus gates. And you can see on this graphic that the Pratt School of Engineering in purple there is interacting really quite comprehensively with the Trinity College of Arts and Sciences and all its sort of affiliated science departments. We have the Nicholas School involved. We have, of course, also involved the Duke School of Medicine. We have 6 signature research areas. And again, if you look on our website and you look at the faculty listed with each of these 6 areas, then you will see that most, if not all, are actually also cross-listed in other areas. Again, exemplifying the cross-disciplinary nature of our activities. DMI also is a host of an educational opportunity in material science and engineering. It hosts, led by Professor Adrian Stiff-Roberts, a growing program that is granting master's and also doctoral degrees in material science and engineering. Some notable metrics from just the last year: we have over 60 faculty, we have over 250 publications in that year, we have a research budget of about 40 million awarded just this year to DMI faculty, and several centers, national or international centers founded here at Duke. If you're interested in the activities of DMI, I encourage you to sign up on the website and then also maybe subscribe to the newsletter where you can see some of these sort of glimpses here over the last semester to stay up to date with what's going on in material science. But now it's really my pleasure to introduce FLI's session, Nurturing Nature, Improving Health with Biomaterials. Today, we'll hear from Duke material scientists, bioengineers, and surgeons who explore a wide array of biological phenomena that unlock discoveries that ultimately lead to new bio-inspired materials that can help the body heal faster and better. The order of the speakers we chose from more molecular tissue engineering towards materials engineering, and then all the way to the clinical implementation. And I'm thrilled to welcome Tatiana Segura, Matt Becker, and Malcolm DeBon. Let me introduce the speakers briefly. Tatiana is a professor of biomedical engineering in the Pratt School of Engineering, and with secondary appointments in neurology and dermatology. Her research is broadly focused on the design of biomaterials for delivering genes, proteins, and drugs that promote endogenous repair and reduce inflammation of tissues. She received a PhD in chemical engineering from Northwestern. She did a postdoc at EPFL in Lausanne, and she joined the faculty at Duke in 2018 as a full professor. She has numerous awards to her name. For example, she's a fellow of the American Institute for Medical and Biological Engineers, and she got development grants also from the American Heart Association. Matt Becker is the Hugo L. Blomqvist Distinguished Professor at Duke University. He holds appointments in chemistry, mechanical engineering, material science, biomedical engineering, and orthopedic surgery. You can definitely see that he is sort of crossing boundaries here. Matt's research is focused on developing bioactive polymers for regenerative medicine and addressing unmet medical needs at the interface of chemistry, materials, and medicine. He's also very entrepreneurial and the founder of 3 startup companies. He earned his PhD in organic chemistry at Washington University in St. Louis, and then did a postdoc in the polymer division of NIST, that's the National Institute for Standards and Technology. He received numerous awards and distinctions. Maybe most notably, he's a Kavli Fellow of the National Academy of Sciences and a Fellow of the Royal Society of Chemistry. Malcolm DeBaun is an assistant professor of orthopedic surgery in the School of Medicine. He's currently practicing as an academic orthopedic trauma surgeon, and he just told us that he just came out of the operating room just a few minutes ago. And he has a special interest in using innovative techniques for bone tissue engineering to improve the care of trauma patients with devastating limb injuries. Malcolm attended Stanford Medical School, and he is now completing his orthopedic residency. He completed his orthopedic residency at Stanford, joined Harborview Medical Center for his orthopedic traumatology fellowship, and just recently joined Duke. So without further ado, please welcome Tatiana Segura.
Thank you, Stefan. If you stop sharing, and I can share my slides. Awesome. Okay, so I trust that all of you can see my slides correctly here. I'm very excited to be here to share with you some of our work on designing materials that we believe can be used as drugs to help our body heal. And so just to start sort of motivating what my lab works on, this is my lab, the Segura Laboratory. And so you can think of normal tissue. If you have an injury of a wound, what we try to do is design materials that we can add to these wounds to then help the body promote healing, and at the end, form normal tissue that hopefully is scar-free and fully functional. And so what is it that we do? How does it work? So what we do, when we simply put, we develop these semi-permeable materials that we can be used inside the body to heal damaged tissue and improve functionality. We are at the core at a biomaterials lab. That's what my lab is. It's a biomaterials lab. What are we inspired by? Well, we are inspired by the ability of our body to heal and repair, and sometimes even regenerate tissue without a scar. So how our body actually does this helps us design new materials and strategies to create stronger functional materials. And so we really try to understand the biology of the different tissues that we're trying to promote functional repair so that we can design better materials. And so this is what inspires and what it is. Now, what motivates us is really the patients and the diseases that we're trying to come up with material therapies for. So one of them is stroke-induced disability. There's very few current treatments for stroke, and we try to come up with materials that we can actually place in the brain after stroke so that we can promote recovery. Poor healing wounds is another major thing we're trying to address. And so there's so many wounds that just don't heal properly. And so we want to develop materials that can actually improve this type of healing in skin. And of course, scarless healing in both stroke and in the skin. There's really a big problem. You have a lot of scarring, you don't have functional tissue. And so we have succeeded some of these things in some of these projects. If we use a stroke model, for example, if you have no material, you see very few regeneration. But when you add a material that we have engineered to promote revascularization and modulate the immune system, we actually see substantial repair of the brain tissue. Another example here is from skin. If you see, again, a material with no scar, this is how the skin wounds will look in a mouse. And then you can see we have no material in here. You have a scar, and that's very flat tissue that gets formed. But then if you add a material, you can all of a sudden promote regeneration. And basically, we have now come up with some features of materials that can actually help the body promote hair follicle formation, for example, in this case of skin. And you can see these are the new hair follicles that are forming right in the middle of the wound. And so what are our strategies? How is it that we do this? Well, we take a look at what is happening after a wound occurs or an injury. You have a rapid immune response. Then your tissue momentarily turns plastic, meaning it attempts to repair itself. And then this window of plasticity closes, and then no further regeneration is possible. You have a scar, and basically you have plateaued, and you can no longer improve in terms of repair and regeneration. And so what we try to do is to do three things. One, we try to tweak the immune system. We try to extend this window of plasticity, or we try to reopen this window of plasticity. And so when you think about tweaking this immune system, what we want is to basically come up with materials that when you first have an injury can lower down the inflammatory response and lay down a foundation that goes through the course of the healing process. Sort of like a vaccine that if you can deliver it, it can act later, right? And so we try to come up with materials that have long-lasting immunological effects. And we've done this in different ways to achieve the regeneration that you observed in the previous slides. The next approach is to extend this window for cellular change. Basically, this is simple. We already have this injury, this plasticity-induced injury processes. How do we extend them? And so this is what we introduce angiogenic materials, meaning creating new blood vessels. How do we create new synapses so your brain can actually function better? And so this is meant to extend that window so that your body has a chance to actually heal properly. And the last thing is probably the most complicated, and something that we really need to start doing more of. And that is, how do we actually open up this window of plasticity after it is closed? So many patients actually end up having a disability much, much later, and you will have to reopen that window to be able to treat that injury. And that might be inducing another injury, or really trying to reactivate these pathways without an injury process. So again, what we try to do is to, once you have a wound, is to implant a biomaterial that will then improve repair and regeneration. And so our current projects are very diverse in the lab. We go from, as I said, brain repair, trying to understand how we can actually switch this inflammation. And you can, of course, read a lot about all of these projects in our website right here. And so today I thought I'll tell you a little bit more about one of our materials more specifically, just so that you get a feeling for how we design materials and how we think about them. And so this material is a MAP, and it's basically, how do you make liquid porosity? Okay, so if you wanted to make a material that is injectable yet porous, how would you do that? And why is this important? So why is porosity important? So when you think about polymers, if maybe you've never thought about polymers, but polymers in solution are basically like strands of spaghetti that are coiling themselves. And if you fix them in space by maybe making some linkages between polymer chains, the size in between these polymer chains is going to be in the order of magnitude of the polymer, which means that if the polymer is in nanometers in size, which it is, this mesh size, or the size of these openings, is also going to be nanometers in size. And the problem with that is that if the mesh size is this small and your cells are this large, how do you actually get a cell inside this polymer? And this has been actually a conundrum that you have in biomaterial science. And of course, we have ways in which we can fix this by, for example, making materials that are remodelable. So they're protease degradable, so your enzymes can actually degrade it, or they're hydrolytically degradable, or they're stress relaxing. But also you can make porosity, meaning you can make the openings larger so the cells can actually come in. So what happens when you actually have a mismatch in porosity when you implant these materials is that if you have a non-porous materials like I just showed you that has nanometer-sized openings, immune cells tend to sort of accumulate at the interface of normal tissue and the material because they cannot infiltrate fast enough. So they try to eat it. They cannot infiltrate, they cannot penetrate it. And eventually this is going to be eaten up, but not really regenerate the way that we want. And so you can see also here that the cells, the tissue sort of tries to grow around it, but you have limited infiltration. And so what we have decided, or what we thought about, was what if you actually make hydrogels that look like a bag of oranges? So if you think of oranges or limes in this case, you could actually imagine that there are spaces in between these oranges, that if you make these oranges the size of a cell or larger, the spaces are going to be large enough to accommodate these cells. And so this material, you can see here, basically there are these beads. And these beads, when they pack, they leave behind these pores or these openings that we can then use for cellular infiltration. And so if you're interested in reading more about these materials, I'll direct you to this publication. But basically, in the tab here, this video is showing the previous material, that is just polymers interlinked with each other. We have now then decided to make the same gel, but now into beads. And these beads now are going to pack and leave behind these openings that allow for rapid cellular infiltration because they're large enough. So if you match the size of these pores to the size of the tissue that you're trying to infiltrate, you actually get much better repair and regeneration just by having this porosity. And because they're beads, they're actually injectable, which is really important for our brain work. And so what is MAP? Well, MAP is a meal hydrogel microparticles. And so they still come out with the same polymers. So we use polymers that are derived from nature. So we have hyaluronic acid, for example, which if you read in many of the beauty products, they all claim to have hyaluronic acid to help your skin look radiant. And so we use this type of materials to actually make hydrogel microparticles using microfluidics. And you can see examples here of our particles. And here is an example of a stable gel. So if you take these particles and now you interlink them with each other, you actually form a stable scaffold. And so what is so unique about this material is that it's injectable, because the particles are smaller than the 30 gauge needle. They have interior porosity, so cells can actually infiltrate through the material. They have exterior porosity so that the nutrients can actually flow in. And if you plant them, for example, in the brain, you see that there's rapid cellular infiltration into the material. And you can introduce a lot of heterogeneity. So you can imagine that in a microliter of this material, you have 1,000 microgels. That means you could have 1,000 different things you could introduce into the material to really accommodate this growing complex tissue. And so here is some movies just showing you cells growing in this material. Now in the laboratory, this happen to be neuroprogenitor cells. And so if you add neurogenic peptides, the cells tend to spread. You can see them sort of growing through the material, actually moving the material. If you don't add any neurogenic peptides, the cells actually end up growing in neurospheres. And so by changing the properties of these materials, we can achieve the same cell type could have very different phenotypes. And this, of course, we think is going to have major implications in vivo. So when we implant these materials, we actually show that cells from the suprarticular zone, which are stem cells, can now infiltrate our material. And you can see them all in green here. And here is, again, a different model of stroke. You have the stem cells, and you can see that they can actually infiltrate the material. You can now see here also that we can promote angiogenesis, and so you can see here the blood vessels going inside. And so I will just end by saying thank you to my lab. And I'd like to introduce Matt Becker, which he's going to be telling you many different things. Yes.
Thanks, everyone. And I will try to move this along and stay on time. And so my name is Matt Becker. I'm in my primary appointments in chemistry, but I also have joint and secondary appointments across Pratt and the School of Medicine. It might be useful for the parents that care that, we actually do teach undergraduates and graduate students. So in chemistry, I teach honors general chemistry to freshmen. And so if I'm torching your child, sorry, your money is hard at work. And in MEMS this fall, I'll teach for the first time in coordination with the DMI, an Intro to Polymer Science course that's part of a larger initiative in material science. And then I also teach graduate courses in polymer chemistry at the master's and PhD level. I've only been at Duke a very short time, so maybe it's a little of interest to tell you a little bit about what we're doing. translationally, I moved here and moved my entire team of about 30 people here 2.5 years ago, right before the pandemic, due to Duke's really focus and value in materials and translation. And so my group broadly is really focused on trying to develop what we call translationally relevant chemical methods that allow you to place bioactive species on polymers. Could be drugs, could be proteins, any number of things. And so we focus very much on each of these clinical areas and preclinical areas here on the left, but we're one of the few groups in the United States for sure that will take you all the way from molecules and really what I think is elegant, of course I think is elegant, small molecule chemistry, all the way to being able to hand a surgeon a widget. And that's one of the things that I think is really compelling about Duke. And just really since arriving here in 2019, these are kind of 6 new areas of research. I'm going to tell you a little bit about our patient-specific printing here in a minute. But we've been really fortunate to start really productive collaborations in trauma surgery and rib fracture repair. We've got a very committed effort in limb salvage and warfighters that's funded by the military. We've done a lot with plastics and hernia and soft tissue repair. Another project that's we've had a long-standing commitment to in non-opioid post-surgical pain management, which is a big problem in this nation. And more recently, and we're excited to be a part of some real growth in Duke's research capability in vascular engineering and tissue engineered vascular grafts. But today I'm going to tell you a little bit about our work in patient-specific printing, and I promised the team that I wouldn't show too many bloody photos, but I'm going to show you one. And so this really motivates all of our work, and I think this is the future of where patient-specific printing is going to really arrive on the scene. And this is the reason I came to Duke. And so the young man here on your left is a wounded soldier. He was shot in the face. You can see that it's a pretty traumatic injury. And there's been a lot of heroic work just to assure that this person can actually eat and breathe and survive. And so probably the thing that's changed the most in my career since I graduated from Washington University in St. Louis a long time ago is that the amount of imaging and digital design has really evolved commercially. And you'll see any number of printing companies out there. And if you look at the modern literature, and even those that are in private practice are seeing this. But the challenge has been this only works when the optimal materials are available. And so I'll swap that here so that nobody gets sick. You might imagine that this is brand new technology, but I'll point you to this paper by my former colleague and still collaborator, David Dean, who was one of the first people to do this. And this is not a misprint. This is in 2003 in neurosurgery at Case Western, where they were actually designing non-degradable implants for traumatic bone injuries and cranial accidents. And the challenge is not that the imaging or conversion technology and print technology hasn't been there, but the materials that engineers and surgeons and clinicians have available to them were never designed to fit this type of printing modality. And for the folks that are curious about the economics, preoperative manufacturing in this space will save a surgery like this between $150 and $300 a minute. And so when Bob Ratchison started this at Case Western a long time ago, this is a 16-hour surgery that involves 3 surgical teams of between 6 and 8 people. And at Case and most likely at Duke, you're looking at a couple of $1,000s an hour just to stand in the operating room. And without running you through all the math, for those of you that it's late at night or early in the morning, the surgery here is 8 to 10 hours faster with 3D printing. And that's one benefit economically, but the fact that this patient is not under anesthesia or under anesthesia for 8 to 10 hours less is really significant. And so many of these polymers you might be familiar with if you've suffered through organic chemistry or been familiar with this field. But one that we're really excited about here is polypropylene fumarate. And we think that has a lot of clinical promise that I'd like to show you some areas where we think this could really have some impact. And over the past, really since 2018, we've developed an entire family of printable resins. And just to give you a little frame of reference here without going into too much detail, This is the base structure of this material. Its degradation products are fumaric acid and propylene glycol. And the purple units here are the ones that are actually cross-linked to form the solid network from the oligomeric resin. And we've done a lot with N-functionalized materials where we're tethering growth factors. We've done some prodrugs. We've done a lot with gels. And we're actually starting to work a little bit with Tatiana on some areas in her MAP particles that I think could be really groundbreaking. But today I'm going to tell you a little bit our elastomeric work. And so again, if you're familiar at all with 3D printing, many people have this idea that it's either bioprinting, which is a very soft gel that people are pre-printing in tissues, or something that's rock hard, like you would use in bone. But today, I'm going to tell you a little bit about the work in elastomers and something that we're really excited about. And so we're really fortunate that we're using and partnering with Carbon. Carbon is one of the probably the 3 or 4 largest printing companies in the world. They're probably most famous for their shoes, but we've actually got the only open materials license in the world with a Carbon printer. And we've been developing resin technologies that actually work. And in some of our bone work that we're doing with Malcolm, who you'll hear from shortly, and some of his colleagues in orthopedic surgery, we're looking at how do we print structure that might recapitulate bone. But this is a fast thiolene-based printing that works up to 70% solids. And when you cross-link this, the ratio at which you cross-link it determines the rate at which it degrades. And if anyone's interested, I've got information at the end that you can dive into this. And so the nice thing about it is that unlike many materials, there's no yield point. So this is a common stress strain curve that shows you a variation in the length of the cross-linkable units versus the lengths that aren't available or that span the cross-linking units. And you can see that can be tuned widely. Also, if you determine or vary the cross-linking ratio, you can actually determine the rate that this degrades. And most importantly, there's no hysteresis. And hysteresis is something that's related to fatigue. And what this cyclic test tells you out to 80% strain is that if you strain this repeatedly over time, that you'll have the same elasticity you have. And just to convince you that this can actually be stretched nicely, here's a short video where I've got a graduate student actually stretching one of our printed structures designed for rib fracture. And you can actually see that you can get 300 or 400% strain before it breaks. And it's actually quite tough. And that video in particular, and a couple of beers, led to a conversation with a colleague at Stanford, Jody Simone, who was actually one of the founders of Carbon, where him and his colleagues at Stanford were interested in revolutionizing what they could do for vascular bypass surgery. And so again, this is the only bloody photo that I'm allowed to show. But in a vascular bypass, they typically strip a vein from a person's leg and they graft that into the arterial system. And the challenge with that is that mechanically, those two grafts are mechanically mismatched. Veins lack the muscles that the arterial systems have. And so Jack Boyd, who's probably one of the best surgeons in the country, has come up with a sheath. And so this is a 3D printed sheath that goes around the outside of the artery that's designed to support it mechanically for 4 to 6 weeks and then degrade over 12 weeks. And during that time, the vascular system is actually able to begin to remodel. And the clinical outcomes, at least in their sheep and rabbit models, are actually quite promising. And so what you see here on the right these are the actually 3D printed sheaths. They have the design support structure here. But when you have an unwrapped system, you might get lucky. And if the arterial pressure is low, particularly in most females, then you'll have a gradual remodeling of that vein into a more arterial-like structure. But very often, if the pressure is high or it's at the graft points where you're actually suturing this, you'll get an aneurysmal structure, or it will actually close and calcify and stenose. Where if you have this structure wrapped, so far it looks like we're getting very phenotypic remodeling from veins into artery. And I think that's potentially very exciting, both clinically and commercially. And if you just look here at a little more mechanically, and I won't belabor the point, this just shows both in this 20-to-1 ratio cross-linked and a 30-to-1. So this is more heavily cross-linked. This is lightly cross-linked, that those mechanical properties are changing with time. And the rate of change is critically important to that remodeling process. While it's working so far, according to the computational models developed by Alison Marsden and her colleagues, the flexibility in the 3D printing structure is really what's enabled this to advance as far as it has. And just really in conclusion, again, because it's a short night, if you look at this plot, and I like to show this because this is a log plot of tensile strength and Young's modulus, and these are all the tissues in the body. Everything from vocal cords, which are very soft and hydrogel-like, to things like tendon and ligament and bone. Most degradable polymers that people are familiar with fall 1 to 2 orders of magnitude outside that regime, so it really reinforces the concept that these materials were never designed to recapitulate some of these tissues. And if you look here at the bottom, the orange and green colors, hopefully nobody's colorblind, are actually our degradable resins, and the black and blue are actually the commercial resins that are out there. And so many commercial resins that are elastomeric are designed to be tough. You see that here. Certainly degradable materials can't reach that. But in the regimes where these other materials are designed to be degradable or not, these are non-degradable resin systems, we are blanketing that resin. And so it's early but exciting. I really want to reinforce that these are fully degradable implants, unlike many other systems that are out there. And we think we're going to be able to cover a wide variation in tissue. And only a couple of years in, we're pretty excited. And so this is my team. We moved a big team to Duke. The folks in red are the one that did the work. It was actually funded very early on by Carbon and also the NIH. And so if anyone's interested or has more questions, this is my email address. You can also find me in the Chem, BME, and MEM site, and we do have a Twitter page. And so with that, I'd like to introduce my colleague, Malcolm, who I think is about 6 months into his tenure now and has really got some exciting things going on.
Thanks so much, Matt. That was great. Good evening or good morning, everybody. My name is Malcolm DeBond. I'm one of the orthopedic trauma surgeons at Duke, and just finished my training and really got thrown into the fire here in loving life, taking good care of patients and trying to do a little bit of science along the way. So this title of this talk is Bioactive Membrane Improves Healing in a Preclinical Non-Union Model. And here are my disclosures. So here's our problem. This is a problem that I deal with every day. So patients come in often with very devastating injuries, and some of these injuries have critical bone loss. So these fractures where there's missing bone becomes a really complicated issue. And it affects 2 million patients globally and has a pretty big market size of $3 billion annually in the US. And unfortunately, there's no great solution to critical bone loss. Right now, one of the ways that we address this surgically is through the so-called Masquelet technique, but it requires multiple surgeries and it requires autograft harvest, which has significant donor site morbidity to the patient. So what we wanted to try to do is we wanted to engineer a solution. So a bioactive implant that is capable of healing critical bone defects without autograft harvest that can be performed at a single stage procedure. So we developed a preclinical non-union model initially, and then began testing our implant. So we first created different microspheres that have osteoinductive, that's growth factors that basically stimulate cells to produce bone. So we had a bioactive membrane made of type 1 collagen and put these different microspheres that contain growth factors into them that degrade over time. And then we put that membrane that was synthetically created around a scaffold that has some strength and compression. And again, this is a 3D printed scaffold. And then we first tested it in an in vitro model to determine if the growth factors would release in a controlled fashion over time. And then we tested it in an in vivo rat model. So here's our in vitro study. It basically just demonstrates that BMP2, which is a really important growth factor for bone generation, and platelet-derived growth factor, which is another growth factor for bone, was released in a controlled fashion over time locally into the environment in vitro. So then once we established that model, or that proof of concept, I should say, we did a in vivo preclinical model of a number of groups. So we had a negative control, which is basically a segmental defect in a rat femur that is unable to heal on its own. And then we had the positive control, which is what we do in surgery, doing the Masquelet technique, which is basically essentially putting in a cement spacer, which you can see here in the intercalary space. And then that forms a membrane from a foreign body reaction from the patient. And then at 6 to 9 weeks, we come back and size the membrane, take the cement spacer out, and put in bone graft. So we essentially did that in a rat. And then we also had our experimental groups, which had different types of growth factor incorporated into the synthetic membrane, and performed that implant and implanted that into a single stage procedure at the time of the critical defect creation. So I'll just show this video real quick. It's important to note that all aspects of the scaffold are covered in this membrane. So that's the membrane. It has some nice usability properties to it, and the scaffold underneath was created specifically for an 8 millimeter defect. And we did an in vivo analysis using x-rays to determine healing over time, which is what I do as a clinician, when I follow my patients. So we got serial x-rays, and then we had this nice grading system to determine radiographic healing. And essentially, if the bone heals, then you see more radial density, and you start to see the bone develop to bridge the gap. So here's an example of the first week after surgery. You guys can see there's a very large defect here into the femur that wouldn't heal on its own. But what you don't see is that there is a scaffold that has two types of growth factor that is incorporated on the membrane that is in this space. But then over time, the healing process starts, and at 4 weeks, you start to see a little bit more calcification, a little bit more bone growth. You see the bone ends are trying to find its home there. At 8 weeks, it looks a little better, but not quite there yet. Still dependent upon that rod and dependent upon that implant. But then at 12 weeks, you see nice bridging callus all the way from end to end, and that's considered a healed fracture. So here's our histo. Basically, we had different groups that I mentioned prior. And I like to look at this row on the bottom here, and blue is bone, basically. And essentially, when we had BMP and plate-derived growth factor, we were able to see bridging bone between the bone ends, which you don't really see as well in the negative control. You do see with the BMP only. PDGF only, not so much, and the Masquelet just looks kind of disorganized, but may or may not have some bridging bone. So then, when we looked at our micro CT, similar type outcome, but looking at BMP looks like our winner here, both in BMP only and the BMP with the platelet-derived growth factor. Both of those groups healed nicely and had some pretty good bone volume quantitatively. So the strengths of this kind of design was that we use translational methodology. So only FDA approved materials were used because, I want to make sure that the science that I'm doing is potentially going to be able to help a patient that I'm taking care of one day. And then we did in vitro proof of concept, and then the in vivo experiment, we used a clinically applicable outcome measure, which is radiographic union. And again, that's what I use in my clinical practice to assess for healing. And then also it's important to keep in mind that none of the implants that we used required cells. So there's no autograft harvest. There's no manipulation of cells. These are all acellular implants, which is important for translatability through the FDA pathways. So in conclusion, we had established a collagen type 1 membrane with BMP microspheres improves radiographic healing. And our potential clinical application is that we can restore viability to bone that's been damaged potentially. And then also we can incorporate this membrane into a synthetic scaffold that has some strength and compression to potentially offer a single stage solution for critical bone loss in our patients that relies on healing. So with that, I want to just thank my team here. And this is some of the research that we started at Stanford and I hope to continue on, especially with our collaborators at Duke. Thanks so much.
Okay, wonderful. Thank you. Three fascinating talks. And you saw, I think, really the full spectrum in terms of thinking about materials and then seeing them applied in patients or at least also in lab animals. So I think this was pretty amazing. So we have several questions. One was, and that's a question for all of you, what is really the timeline for the development of some of these solutions which we've seen today? I mean, if you think about concept to human or clinical use, does it take months, years, tens of years? So what is your experience?
Well, I haven't gone to humans yet, for sure. Although I did co-founded a company to develop, to utilize our materials for skin regeneration, and the clinical trial for that starting next year. So that's a lot faster. I think any of our brain work, we have to do more work because it's, pretty delicate. And I think that it will require more testing. So I would say from initial inception, something like that, it will be decades before it's actually be able to use. But I think other technologies for like skin is a lot easier to get to humans.
And just where we are, Stefan, I think we're probably a year or two away from our first in man for this particular technology. So, and it's really about risk management in areas where current things don't exist at all. I think the tolerance for risk is higher, but in things where you might be impacting multiple systems, certainly the regulatory agencies are much more cautious.
Malcolm, do you have anything to say about your material?
Well, we're just getting started. The next step for us is going to be a larger animal model because it's hard to heal critical defects of a human size, right? You can say you can heal something in a mouse or a rat, but the critical defect in humans is more voluminous. So we need to make sure our methods are able to translate into a larger animal model. So that's the next step for us.
Thank you. So I have a question for Matt. So when you talked about your arterial remodeling, you basically made a point that it's really the mechanics of the material which is really critically important. To what extent can you also use or combine this with biochemical cues? I mean, you could also essentially load your constructs with essentially pharmaceutica or I guess biochemical cues to sort of get this sort of down the right pathway.
Yeah, no, absolutely. And we're looking at anytime you impart biologics into a material, you run into a combination product. There's just a higher level of scrutiny of that as you kind of march through your GLP and preclinical trials. And so we are certainly doing that in my research lab. But I think the proof of concept out there, especially in this vascular bypass arena that we're working with Stanford, is that the complication rate is so high. They're really looking for anything that will knock that down to any extent. And I think, as that advances, everything that's more more bioactive, I guess if I want to call it that, will march behind it in short order. But it's really a risk management approach, and can you help patients now versus what might be perfect, 5 or 10 years from now.
So we have a question from the audience, and it's any published animal or human stroke trials?
Yeah, definitely. We have many publications using our materials in rodent stroke models, but not human trials. And so if I'll put something in the chat, that's for example, an example. But if you go to my lab website, all the publications will be there, and you can actually search them for stroke, and you should be able to see all of the different things that we have done.
Thank you. So I have a question also for Malcolm. When you look at sort of your bone repair, I mean, these are devastating injuries which you're trying to repair. So how important is actually mechanical stimulation for the healing and the growth?
Yeah, it's critical. If the bone doesn't load, according to Wolff's law, it doesn't heal. And so it needs the implant needs to have some type of mechanical stability associated with it. I do augment all my implants, implanted grafts with fixation. So you saw that even then that rat model had a nail that went through it. But ideally, the implant would be able to sustain load and be able to function really as an intercalary bone graft.
Thank you. So, and a question here for Tatiana. So I was really intrigued by your, I guess, orange model of developing a scaffold. And what really struck me was, can you make that actually in gradients? Or is there any value to make it as a gradient?
Yes, definitely. So because it's I didn't want to get into these granular materials, but once their particles are jammed, basically the forces in all the particles are together. It behaves as a bulk unit. So you can definitely load a syringe in a gradient fashion in different types. And when you inject it, they will come out like that. So you can actually create, even in the stroke, actually different regions like that go radially. And it will be very important because everything in biology is about gradients. So cells move towards a high concentration of different types of growth factors or chemokines, cytokines. So it's all about gradients in biology. So definitely it's very important.
Very cool. So there's a question from the audience. The viewer states, your work is all fascinating. Are any of you active in the area of neuroregeneration?
Yes, for the stroke project, it's all trying to promote a new brain tissue and new neural connections. And I think Matt mentioned some peripheral nerve injury work.
Matt, do you want to say anything more about that? Or
yeah, we've been looking at artificial conduits designed to span large gaps, using a tethered. So rather than using whole growth factors to create these gradients, one of our areas, really more fundamental or basic science at this point, is looking at small protein subunits and how concentration profiles of those might be used to induce different, biased migration in different cell types. So again, early, but I think promising.
Cool. So this is maybe a sort of more of an insider question, but I was particularly taken by Matt's Ashby chart, which is essentially the log-log plot of mechanical or of properties, material properties, which you showed towards the end of your talk. And this is actually really the first time I've seen this in the context of biomaterials, which is probably just showing that I really don't know that much about it. But is that sort of a common design tool or what bioengineers use to think about the universe of biomaterials? I mean, it's quite commonly used in material science.
That plot is a summary of about 1,500 papers that was compiled when I was really mad at a student for failing one of their preliminary exams. Conducted that, but I think we sent this. My colleague and I sent this student on this adventure. We'll call it an adventure. And what they came back with, we were stunned that in fact the log plot of that so lined up so linearly. And I think there's probably a evolutionary component to that's probably too brilliant for me to figure out. But I think when you start looking at the materials that many folks have used especially in regenerative medicine, to try to meet some of these needs, they're not anywhere close to that line. And I think the challenge there isn't that they won't work, is that there's other materials out there that people are only beginning to explore that might work better. And I think for us as synthetic chemists, especially here at Duke, in an environment full of one of the best biomedical engineering programs in the world, we're excited to be here.
Great. So we have one more question. That's going to be our last question. It's been 1 million years since I was in school, what is the life expectancy of most implants these days?
So, it depends on the implant, probably. I'll just speak to me. If everything looks like a hammer to me. So for orthopedic surgery, for total knees and total hips, for example, that implant, it's not a biomaterial, is metallic. It's about 20 years or so. But what we're really trying to do is initiate the healing response such that our implants regenerate tissue and last a lifetime.
Thank you, Malcolm. So I guess with that, let us thank again these three outstanding speakers, Tatiana, Matt, and Malcolm, for fascinating talks and hopefully fodder for thought, future thought. And again, just look at their websites, look at DMI's website, see what exciting things are going on at Duke in the material space. Thank you.
Thanks so much, Stefan, and thanks to all our wonderful speakers, Matt, Tatiana, and Malcolm, for really great presentations, and of course to our audience for joining us. Today's session was recorded, and it's going to be posted to the Duke Alumni Lifelong Learning YouTube channel for future reference or to forward to other folks. Our next session is going to be March 29th, and we'll explore the accelerated materials discovery. For more opportunity, check out a robust course syllabus, and we look forward to seeing you at the next Forever Learning Institute. Thanks so much, all.